introduce our next segment, a rapid fire academic therapeutic development talk. So four of us, you know, I gathered some people representing what the academic sector can do, because part of the symposium is also how do we harness reserve capacity, and particularly for SARS-CoV-2 drug development discovery, because it's classified as a BSL-3, there's actually not so many ways the private sector can innovate as rapidly as the academic sector, because we had the labs, BSL-3 labs, that, for example, Gilead didn't have. So I'd love to introduce to you several people today. I will start with Dan Nomura, who is a professor of chemical biology in the departments of chemistry, molecular cell biology, and nutritional science and toxicology here in Berkeley, and is a real leader on discovery of new chemical entities, particularly covalent mechanisms of actions, which are great for inhibiting, for example, viruses. Also co-founder and head of the scientific advisory board of a company called Frontier Medicines, and also the director of the Novartis Berkeley Center for Proteomics and Chemistry Technologies, focusing on using chemoproteomic platforms to tackle undruggable targets. All right, next up is Professor Anders Näär, who's professor of metabolic biology in UC Berkeley in the Department of Nutritional Science and Toxicology. So Anders is originally from Sweden and worked with Bob Keegan before as a postdoc, and he's specializing, or what he's talking about today, is antisense technology, which is has, you know, the mRNA technologies have gotten a lot of press because of the vaccine. Of course, it's a great potential to be highly adaptive and very fast in the response to new strains. So it's an interesting technology, different from the usual small molecule approach. So take it away, Anders.
All right, thanks, Julia. Thanks for the invitation to present here today on our therapeutics efforts to target SARS-CoV-2. Can you see my screen?
Yes.
Great. So as Julia alluded to, my lab pivoted back in March last year to apply our extensive knowledge of targeting therapeutically microRNAs and other RNAs using antisense technology, state-of-the-art antisense that binds to the RNA and either targets them for degradation or acts via steric hindrance to prevent the RNAs from working. I'm actually going to start with another approach that we also pursued in parallel in collaboration with former colleagues and collaborators at Harvard Medical School, as well as a partnership with Google. I'm going to start off with that, actually. I haven't talked about this before. So it's a structure-driven drug discovery effort using ultra-large virtual libraries to screen billions of small molecules against a particular target. And we can, it's basically target agnostic. We can target anything. And in this case, we targeted every single structure that had been solved on the virus using this in silico approach. And I'll go into some detail on that. And the second part of the talk will be delving into our RNA medicine approach, as I mentioned, using therapeutic targeting of the viral RNA. So the first approach was really inspired by some groundbreaking work by my former colleague, Hari Arthanari at Harvard Medical School, and his postdoc, Christophe Gorgola. So they actually developed a high throughput screening methodology using hundreds of thousands of computers running in parallel that can screen up to 1 billion compounds against any particular structured target. And they actually get nanomolar affinity binder compounds out of this type of methodology, even targeting protein-protein interactions, which are really thought to be undruggable, and allosteric sites and so on. So there was this really groundbreaking discovery that was published last year in Nature. It was a 10-fold larger screen than had ever been conducted before. And so I discussed with them the possibility of applying their technology to SARS-CoV-2 crisis. And we decided to tackle this in collaboration with Google. So Google provided the computational power, if you will, and $1 million in computational credits. And we screened over about 2 weeks against every single structure of the SARS-CoV-2 virus, as well as the ACE2 receptor itself. And so there are lots of different structures that were solved very, very quickly in a matter of months after the virus sequence had been published. And so that enabled this very large drug screening effort. And just to summarize, we actually screened 50 billion compounds against 17 different targets, more than 1 billion compound against each target, against both viral and host cell targets, as I mentioned. So including ACE2 as the receptor for the spike protein that's on the surface of the virus, the spike itself. So this is now a protein-protein interaction that we're targeting. And as I will show in a moment, we were very successful in targeting this protein-protein interaction, as well as what Dan mentioned, viral proteases such as MPRO and PLPRO and so on. And we found that we can actually target many protein-protein interaction interfaces, as well as allosteric sites that have previously been considered undruggable. And now in a secondary screen in my laboratory, as well as with Julia Schaletzky and Sarah Stanley's BSL-3 lab at UC Berkeley, screened 5,000 hits from this 50 billion compound campaign. So basically the top few hundred hits from each of these individual screens. We screened in 2 different cell lines that are commonly used to query viral replication, so Calu3 and VeroE6. And we found that a number of compounds validated with low micromolar efficacy in these assays, as I will show you in a moment. And Hari's group at Harvard Medical School also validated that these hits were all nanomolar hits on target. So basically a very similar activity as what Dan just mentioned. And we see that for every single target. And this is still unpublished. So just to show you an example here, remdesivir, you all know about remdesivir. It's an inhibitor of the RNA-dependent RNA polymerase. And it doesn't work very well in these assays. So many of our compounds, hits from the screen, actually work better than remdesivir. Some of them are against MPRO, for example, that Dan mentioned, nanomolar inhibitors, PLPRO, but we also found these protein-protein interaction inhibitors targeting the spike interface or the ACE2 interface with spike. And just to show you here that we have, this is starting with a structure, so we know exactly how the hits work from the get-go. So they bind in these interfaces and we can interfere with this protein-protein interaction very effectively. So that's quite a stunning advance, I would say, in a very short period of time. So all of these efforts were carried out over about 4 months from beginning to end. So starting back in April, we were done by July, which is quite striking, and with the majority of the screens. And then we submitted a paper, I think in August or September. And so that was the first campaign. And I think this is really telling us that we are able to apply this computational power now in new ways that enables us to find small molecule hits against what was previously thought to be undruggable. And this opens up numerous potential therapeutic targets, not only in viral diseases, but in many other types of diseases as well. We could also possibly repurpose some of these drugs for other coronaviruses or other viral diseases down the line. So that's the first part of the talk. The second part, I'm going to quickly summarize data on the antisense approach that we're taking to target the virus. So antisense oligonucleotides are basically a molecular Velcro. These are short oligonucleotides that bind in a Watson-Crick base pairing manner. So they bind to the messenger RNA or any type of RNA with very high affinity. So this is very different from the traditional small molecule targeting that I just mentioned. They're very, very high affinity and efficacy, and they can be tailored even to specific mutated DNA and RNA, and they're typically well tolerated and low toxicity in vivo. So the type of antisense technology we have employed is called locked nucleic acid antisense. It has this oxygen bridge moiety here that locks the base into one conformation, and this affords much higher affinity and stability. So it's much higher affinity than regular antisense oligonucleotides. Another chemical modification is on the backbone side. So instead of a phosphorothioate backbone, we'll have a phosphorothioate backbone. And that also affords greatly increased stability and pharmacokinetic properties on the oligonucleotides. So this is heavily chemically modified azo. And we apply these to target spike. We target the human ACE2 receptor, as well as other sequences within the virus. For example, the 5' leader sequence that we'll get to in a moment, the 5' UTR, as well as the frame shifting element in the virus. And I'm not going to talk about these in detail, but a number of different regions of the virus were targeted. The one that worked really well is this 5' leader sequence. So this is a sequence that appears to afford increased stability to the RNAs, and it's actually appended onto all the structural RNAs in the 3' end. And we're not exactly clear how that happens or what the function of this 5' leader is, but it seems like it's programming increased stability of the viral RNA. We found that antisense targeting of this 5' leader is incredibly effective. So if we introduce the antisense into human cells and infect the cells now with a virus, we find that we even see single nanomolar IC50 efficacy in decreasing the viral RNAs, as well as in the production of viral particles, so infectious virus. So very, very potent antisense. Then we decided to test this antisense in mice, and we found that what worked surprisingly is simply intranasal introduction of this antisense just in saline, in salt water, into the nose. So intranasal delivery, once daily. So we can dose before as well as during the infection. We can dose before and after, and we still see efficacy. And with this paradigm, we see about 100-fold decrease in the virus in the lungs of the animals, both by viral titer as well as by RNA levels. So it's quite striking decrease. If we use RNA-seq analysis, we look at the transcriptome, what happens in the lungs, we see that the antisense dramatically decreases the viral load in the lungs as judged by RNA, and you can see that more effectively over here as well. So it's a dramatic impact on the virus infection. If we now do a staining for the SARS-CoV-2 protein N, structural protein, very heavily infection in one lobe of the lung here, in the LNAX treated animals, we see virtually no infection whatsoever. And one concern with antisense and other RNAs introduced into the lung has been that they may elicit immune response, and we don't see any evidence of that. If anything, it's actually less immune response as compared to the control. As all of you know, there is an emergence of variants now that's really concerning. The variants are concerned, the UK strain, there are a couple of California strains and so on, South African strain, and they're spreading worldwide. And we wondered, could the LNA azo, because it doesn't target the spike protein that's mutated in all of these strains, might it have efficacy against all these? And indeed it does. So we see no changes in efficacy with the LNAX against the different variants that we have tested. And that's in vitro, and here showing in vivo. So it's the South African strain infected mice, and we see about 100-fold decrease in the viral load of the lungs of these animals upon infection. So just to summarize, we found a strikingly effective antisense to target the structurally conserved non-coding region in the virus, not spike, and it's active against all of the variants. Simple once daily inhaled treatment in saline, and it's very efficacious, and we don't see any toxicity. Just to summarize, we have developed two different methodologies to target the virus. high throughput computational screening targeting all the different structures of the virus with small molecules, giving nanomolar compounds right out of the screen, as well as very potent antisense compound delivered intranasally. And as Dan said, we don't have any US government funding either. We inquired whether it would be possible to get US government funding, and it was basically it would take 6 to 9 months, which is something we didn't want to wait for, obviously. And we were very fortunate to get about $2 million in funding from a number of different granting agencies, similar to what Dan mentioned. And thank you all for your attention. Happy to take that.
Thank you, Anders, for a fantastic talk. This is also a great story. Are there any questions from the audience? So maybe I'll ask a question. With the like Moderna and BioNTech also pursuing these mRNA approaches, do you see like more activity in this space? Because the locked antisense nucleotides, are kind of a bit of an exotic approach, right?
Yeah, so there are very few actually who have tried their hand at using antisense. And most people probably didn't realize that you can actually have efficacy simply by inhalation in saline. We actually tried to inject subcutaneously, and that didn't work at all, which is the standard route of administration for antisense. And so the real breakthrough came with us just trying kind of on the lark, whether just plain naked antisense into the lungs would do anything. And we were shocked to see this incredible efficacy. So I think this is a new paradigm really in the antisense field. Now there are other companies that are also trying other types of approaches that are kind of orthogonal, like sRNA, small interfering RNAs, and there's some traction there. I think Ver and Alnylam, they have a collaborative arrangement, and they reported some positive data, but I don't think it's made it to the clinic.
Yeah, and I think important is also to compare, for example, for the influenza vaccine, they do a clinical trial in the beginning and then they recertify every year with a basically new sequence, right? And something similar is in principle possible with these antisense approaches, right? If you develop a drug and basically you adapt to emerging strains, you could try to make the case at least to get basically approval based on very similar. You don't have to go through the whole process again potentially. So
right.
that would be an advantage in terms of reacting fast to new variants or new subtypes of certain virus, for example. So there's a question from the chat also. Any estimate of a time to a publicly available drug, Anders?
Well, we're pushing really hard to get this towards the clinic. We're basically getting ready to talking to the FDA in terms of getting guidance from them for what we would need in terms of PK, PD, and tox studies in animal models, and then before we go into IND and human phase 1. So my guess is we could, if everything goes well, be in the clinic in 6 to 9 months, and that may be optimistic, but we're hoping.
Okay, any other questions? Thank you, Anders. Been a pleasure.
Thank you so much.
Yes. All right, so next up, I'm myself. I'll share my screen. And let's see where it is. We worked on a project looking at compounds potentially synergistic with remdesivir. So we had, when I started in Berkeley, I founded the UC Berkeley Drug Discovery Center to allow high throughput screening, small molecule screening infrastructure on campus. And as the pandemic hit, we realized we had two things at the right time. And one of them was the screening ready library and infrastructure in my center, and the other was a biosafety level 3 laboratory that I was able to access through my collaborator, Professor Sarah Stanley, who worked with me on this program. And with this together, we were able to conduct a screening campaign against live virus, which is a non-trivial thing because of the biosafety regulations that we faced with this pandemic. The idea was to do a repurposing screen with FDA-approved compounds because those compounds could, in theory, be prescribed by a physician directly with off-label. There's no approval needed if you do the same dose. We were going to run the assay in Sarah's lab in the biosafety level 3 with SARS-CoV-2 infected cell lines to identify relevant compounds. And then the idea that I have was to enhance efficacy of remdesivir specifically, because we already knew remdesivir was going to play a role as an antiviral, but it's not really working. It's not working that well, as also the clinical data show at this point, but it was already clear in the beginning. So the idea was to do an unbiased screen for synergistic combinatorial therapies. And the way you can do that is to establish an assay in the presence of low concentrations of remdesivir, and then do a screening strategy to isolate compounds that are active in combination. The way most people do this is they first screen separately and then they combine the best ones. But I knew from my work previously that that misses a lot of compounds that are actually only showing up if the other compound is there. So that's why we set it up like that. And we were able to look at cells. We used human lung cells infected with SARS-CoV-2 and then treated them with compound here that inhibits the virus. And if you don't inhibit the virus, the cells shrivel up and die. And that difference you can measure using a homogeneous assay, cell tidal glow readout. And also here the expertise we had from a postdoc from Eva Harris's lab, particularly Scott Beering, and also some other people working on this team was instrumental, because when you encounter a new pathogen, the first thing is you have to actually develop an assay infecting cells with the virus. And this is also not a trivial thing, and not every virus infects all cells. So there's a lot of experimental work to even get a consistent infection and then also see an effect of the virus infection on those cells. And the team was able to do a really good job on that. And we had a really nice assay in a short time frame. So we conducted the screen, and bear with me here as I show you some screening data. We did it in two ways. One was without remdesivir, just straight up compounds, and you see inhibition of cytopathic effects. So all compounds ranking highly here inhibit the virus. And you can see there's actually very few compounds if you just do this without remdesivir, which was consistent with the previous data. Remdesivir is the best of the single-use compounds in this library. But if you combine a small amount of remdesivir with the other library compounds, suddenly you get a good chunk, about 100 compounds that show really strong activity. And without remdesivir, no activity, but with remdesivir, really strong inhibition of the virus. So those are compounds we were suspecting could be synergistic. And we did a dose response matrix experiment where we combine increasing concentration of remdesivir with increasing concentration of the partner drug, and then generate this matrix that combines all the different concentrations of these two drugs. And that can be computationally analyzed, and you can get really high quality data about synergy versus additive effects. And the best compound we found was valpatasvir, which is a hepatitis C NS5A inhibitor. You can see here, this is a three-dimensional plot looking at virus inhibition in red, and valpatasvir on its own doesn't show much activity at all. So you can see up to really high concentration, 40 micromolar. Remdesivir on its own at the concentration tested, also not much is happening. But once you combine the two, boom, activity goes up really high. And that's also true, just in different representations here. And if here's the raw data, basically virus inhibition, this is the derived score that looks at synergy over all these concentrations, and you can see that there is a real bump here of activity, and the synergy score is 85 at the max, which is really high. Everything that's higher than 10 is quantitatively described as synergy. And Here you can see the dose response of remdesivir on its own in these cells. So actually relatively impotent. You need about a micromolar to see much of an effect here. And if you combine it with the valpadosvir, it pushes the EC50 into the nanomolar range. So we have a 30-fold improvement, which is significant. This was a class effect. We see this with all hepatitis C inhibitors, NS5A inhibitors. So I'm not showing that data just because of time, but we have many other compounds. They all show this, although valpadosvir is the strongest. And we were able to confirm this also looking at immunofluorescence here in the bottom. If we infect cells with, and just treat with DMSO, they're very red. That's the virus in red. If you add some remdesivir at low concentrations, there's still a lot of red. Once you combine with valpadosvir, it basically looks very similar to uninfected cells. And we also measured, the stain on the cells quantitated, and then infectious virus particles. You can see valpadosvir on its own, not much effect. Remdesivir on its own, not much effect. But together, they get really strong in inhibiting replication of the virus. And if you analyze by PCR, same result. And the upper one shows valpadosvir, lower one shows elbasvir, which is also another HCV NS5A inhibitor. So we were able to show there's really good, strong synergy in these in vitro assays in cells. And we excited about this because the clinical benefit of remdesivir is limited by the concentration of remdesivir active metabolite in the lung is too low to really matter that much relative to the relatively important IC50. So we think that if we can shift the EC50, we could get a lot more efficacy in the tissue. And also you could treat a lot more patients with less remdesivir, which was a bigger deal earlier in the pandemic than it is now, but it's still also a cost factor, and also reduce probability of resistance development. So currently we're looking at this in animal models and see if we can get enough exposure of valpadosvir to observe a positive effect. And we also work with another collaborator at Columbia University, Junior Ye, and he was able to show with some mass spectroscopy approaches, shed some light on the mechanism. So if you treat with remdesivir alone, you get termination of RNA synthesis, and then there is a proofreader enzyme that counteracts that. We were able to show that our compounds like valpadosvir actually inhibit this proofreader, which explains why you don't see much effect on its own, but once you have combination with remdesivir, it really takes out the virus very effectively, because suddenly the virus begins to rely on the proofreader in order to fix the damage that has been done by remdesivir. So because we want to talk about barriers too, I just want to talk about my experience, and also, we raised $1 million fund to kickstart early research, and also received some feedback from awardees there about what has hampered the work that is clearly needed early in a pandemic to find new solutions. A key element here is the biosafety level 3 designation for this virus. I mean, many of you will know that for diagnostic testing, it was classified as biosafety level 2. Otherwise, we couldn't have had any diagnostic testing really on a certain scale. So for bodily fluids and actual clinical sample, it's BSL2, but if you want to propagate the virus in the lab, it is biosafety level 3. That was probably a very consequential guideline, because other pathogens, they go for biosafety level 2 with BSL3 precaution. You could have done that, but they didn't. So it's BSL3. That means very, very few labs actually have BSL3 capabilities. And about 10 years ago, they did a big effort in the US that every state has at least one. And now every state has at least one, but some states have one, and that's it. So private sector generally doesn't have it. That's an academic thing, but then academic centers don't really have, somewhat limited drug discovery infrastructure, and definitely very limited development infrastructure. So that makes it hard to do therapeutics discovery in a pandemic if you are limited by that. Gilead now has a biosafety level 3, but it took about 6 months to get it through. The process is very involved because it has to be designed a certain way. The floor covering has to be a certain way. It's got to have HVAC with certain turnover rate, and you have to make sure all the waste is completely decontaminated. So it's fairly complex, and local authorities need to be involved. And the people who get to work in the BSL-3 also need to be extensively trained by other BSL-3 lab members. And that has been, I think, almost a bigger pinch in my mind, because if you need 2 months training for someone to really work independently in the BSL-3, and then the people who currently have authorization handle experiments for everybody, because they're the gatekeepers for this resource, they don't have much time for training. So Sarah's lab has really taken a leadership role in collaborating with people across the board and helping them execute on these biosafety level 3 experiments. And this also is something that's, I think, often overlooked in the discussion, but you need people who have access to these labs, also willing to collaborate with you to ultimately tell if the compound you are investigating is actually working in the virus experiment. So that is important, clearly a barrier for research in this area. And then also, lab closures, we have the distance requirements, BUA updates. Every new experiment you're doing, you have to update the biological use authorization, and it takes quite a fair bit of administrative work to do it, and review and response to comments. Maybe it's a little bit of overkill, I have to say. Then, the COVID, I understand, but it's even for, really quite harmless things. We have to jump through a lot of hoops in order to get a new experiment started in the lab. The other recurring theme I've heard from many of our RDs, and certainly experienced myself also, is that the government funding model is just absolutely not suited to effectively support a pandemic response. Despite billions being allocated to our government science agencies, I can't really tell that any of that money trickled through to basic researchers in the universities. Generally, the mindset is very risk averse in terms of what gets funded. It's also way too slow. Even the emergency rapid responses that were proposed took 4 months or 3 months of review, which is as Barry was saying, if you think about exponential growth of a pandemic, every day counts, and you just can't review a $50,000 proposal for 3 months, while at the same time we're losing billions every couple of days in GDP. So it just doesn't even make economic sense. But it's very hard to change the old ways, because that's how we typically allocate funding. And to get a new research project funded, it can take 1 or 2 years of several cycles of fundraising through the government agencies. And in a pandemic, you just don't have that. I also find that decision making is ultimately determined by a small group of experts who are great experts. But for COVID, what we've seen is, I would say 95% of the field now has entered just very recently, because everybody's coming from different areas of research and bringing very new technologies to the game. Very few people worked on coronavirus before. Used to be a real niche thing. So then the question is asked, how do you review that science without biasing towards the existing few experts frame of mind? So it's very difficult to fund anything that's new, that's novel, that's maybe a bit risky, but could work. Also very hard to integrate interdisciplinary and private sector investigators who don't have an NIH track record. So that these are all elements of the funding mechanism that make it really hard to kickstart projects. And that's why CEND also got involved in raising money to get people off the ground while they raise other funds in parallel. So in general, there's lots of time spent fundraising for pretty common sense projects in a pandemic, where actually the number of scientists who can even do this work is limited and should be treated more like a valuable resource. So instead we write proposals and do paperwork. So often also the proposal requirements are extensive for a small amount of money, a lot of paperwork. So that's why I loved, I was also a fast grant awardee like the previous speakers, and I really loved that funding mechanism because it was relatively easy, straightforward to pitch, and had a good chance of funding. And in fact, if I look at these projects so far, without the private funding, none of these would have happened, and all of these projects were very, very successful. And then another barrier is lack of access to animal models that need to be developed. Also, those are in the biosafety level 3. So there's a limited number of collaborators. The few people who have these animals have limited capacity. The animals themselves are, basically very hard to get if you need a certain genotype. So these are basic infrastructure elements I just want to bring up that need to be addressed also for pandemic preparedness, because we should make sure that the research is happening quite freely and can inform development of therapeutics, vaccines, diagnostics rapidly, and is not really limited by access to reagents and supplies. All right, and then the last small thing I want to close with is I also have a data science project going, which is, a query of publicly available databases. We had a very simple idea: have people already received remdesivir in these combinations because of underlying conditions? Can we see any effects on outcomes? Very simple idea, but incredibly, incredibly hard to answer because we have no centralized a health record. I mean, that's the number one thing, is a horrible piecemeal. At this point, we've treated millions of people with COVID, and there is data in this data set that could tell us how to best treat these people with the best outcome, but we can't access it, we can't query it. The data sources we have tend to exclude treatment data because everybody is focusing on spatial representation of outbreaks. It's not even, nobody really focuses on treating people, like what works best in the hospital setting. The people with treatment data sets with treatment data often don't have mortality. So we had one case where we saw a different plus or minus drug, but we couldn't tell if the hospital stay was shortened because people were in good shape, or if they left the hospital in a casket. That part was not captured. And then there's also no way to preview the data sources, even what's in it, without actually pre-qualifying, doing an IRB, and going through lengthy contracting and paperwork. And then you go through this, and in the end, you just learn that the data the data set is too small to really say anything. Some require indemnification agreements. We had a 3 months delay at the university because they need to sign. They found some clauses unacceptable. Meanwhile, meanwhile the pandemic goes on, right? Then also some sources require IRB approval for a query of completely de-identified data without any personal information, even for research, but you need to get IRB approval specifically for COVID, not for any other diseases. And also really hyper secure environments for the queries themselves. You need to go through really complicated setup. You can't use all software. Limits analytics, really limits what you can do. This is a bit better in other countries. The UK just started a big EHR data source. The US spent close to $40 billion in the last 20 years on building EHR. And all we've got is a system optimized for hospital reimbursement, and not even for patients, because I personally have at least 4 or 5 of these EHRs, depending on who I'm seeing as a provider. And it all works for them, but it doesn't really work for patient, and it certainly doesn't work for research. So I think that has to be fundamentally rethought as we discuss where to go from this pandemic, because there's clearly a lot that could be done with de-identified data. But if you can't ask the questions, you can't find any answers. All right, so with that, I'd like to thank the team working on this project, Professor Sarah Stanley's lab, Sammy, Claire from the Teach Lab, Livia, Doug, Tom Graham, Eric, Scott, and we got funding from FAST grants, also private funding. Julian working on a data science project, and Tom Graham helped us with the RT-PCR. And we also received some COVID catalyst funding for this project. Thank you so much for your attention, and happy to take questions. If anyone has any. If not, are there any questions? Yeah, how would you propose to accelerate the BUA process? The downside, especially in the case when there's much diagnostic experience, safety should be of primary concern. Yeah, it's true. I think the BUA process could be accelerated just by having committees, meet frequently and be able to give feedback frequently. For the BSL-3 pathogen, I think it makes a lot of sense. But we've also had trouble getting, BSL-1 BUAs turned around in any reasonable time frame. I mean, at some point, I just feel, do you really need to go with every single experiment that you're doing? Do you really need to get BUA approval for every single experiment? I mean, it just limits, if you're a lab who does new things all the time, like we do with the Drug Discovery Center, we hitting, the barrier of that pretty hard sometimes, because it's even, things that are classified as BSL-1, every time you have to go through, and they come back with comments. It's not even like, BSL-1, like I submit something from another investigator that they got it through without comments, I get 5 comments and I have to answer them. So it's a bit, I think it's a bit, it's a whim of people sometimes. All right, DARPA mechanism. Yeah, I would love to. Can you unmute yourself, Barry?
Yes.
Yes.
I'm not sure this is terribly helpful, but my experience with DARPA was quite extraordinary in that, in contrast to having large committees like the NIH of 20 people trying to protect the public's money and not taking risks, the section that I had reviewed for was run by a single guy. He had been a Green Beret behind the lines in Vietnam, and he had total control over the money. Mm-hmm. And he actually wanted to find microbiologists who would be interested in studying the diseases of interest. And he gave them this was years ago, $100,000 if they would agree for any research they wanted to do, if they would be agreeable to advising DARPA on what the opportunities were for new drugs and interventions. So the model for that is the Naval Research Center. And the way it works is if they're successful, that's good. If they're not successful and don't come up with products, they're fired. Mm-hmm. It doesn't work on a committee system. And I had never thought of it in this context, but that's the only way you could get money for drug development quickly, is to have someone smart enough to know who to ask for advice, mm-hmm. and have no constraints on giving the money out the door quickly. And DARPA was able to do that.
Mm-hmm. That's a great idea. I love it. I think the time, element, because like that, you can get at least some money out the door very fast. I felt strongly that in the beginning, there's a need to have a very small amount of money, but if a lab doesn't have unrestricted funding, they just cannot start working on anything related to the pandemic. I mean, so they need a little bit, and then in parallel, they can start developing these grant applications and making them fundable. But if you can't do anything because you're missing 20,000 for reagents, I mean, it's a shame, right? All right. Thank you so much for that input. So last not least, I'd like to introduce Aashish Manglik, who is an assistant professor at the Department of Pharmaceutical Chemistry at UCSF, and originally hails from Washington University in St. Louis, worked on salmonella host interactions there, and then went to Stanford, worked there with Brian Kubilka on GPCRs, so all very pharmaceutically relevant things. And then he's now continued as a Stanford Distinguished Fellow at Stanford University School of Medicine, and then began as an assistant professor at UCSF. And I noticed a work that he had done on inhaled nanobodies that seemed really, really exciting. So I asked him to speak today. Welcome, Aashish.
Great, thanks a lot, Julia, and thanks for inviting me to this really great symposium so far. The talks have just been exceptional, really a great breadth from the basic science to policy. So what I'd like to tell you about today is another example of a real pivot from people that normally don't work on anything related to coronavirus to trying to get a new idea into the pipeline. And that's a really broad collaboration between my lab and a number of other groups at UCSF to provide passive immunization by a new method, which is in an aerosolized format. So again, most of this research happened, because my lab, like many other basic science labs at UCSF, had to shut down in the middle of March. An amazing set of trainees decided, instead of just sitting around, they would try to do something with the technology that they had been using day to day anyway. So at this point, this is, I don't want to belabor SARS-CoV-2 biology, but I do want to highlight one simple thing, which is that in order for the virus to gain entry and take a hold in the first place, there has to be a binary biochemical interaction between the spike protein on the surface of the coronavirus and the ACE2 receptor on the surface of your cells. So of course, by now many of you hopefully have already gotten the vaccine, and those vaccines are designed to provide the spike protein to you such that you can produce antibodies yourself to block this binary interaction. And the cool thing about your immune system is that you develop a polyclonal response of many antibodies targeting many different epitopes on spike as well as other coronavirus proteins. But another strategy to prevent infections, and one that could take hold very early in the course of pandemics, is to simply block the interaction between a viral fusion protein and its host cell receptor. And the idea behind such an approach would be that instead of trying to drive your immune system to develop an active immune response, by providing a blocking reagent, one could provide passive immunity. So this idea of passive immunity is very old, and it goes back as far as the 1918 influenza pandemic. And there's several strategies that one could take for this. The first one that got some amount of traction early in the course of the SARS-CoV-2 pandemic was the idea of convalescent plasma. Now, again, this idea is really quite old, going back almost 100 years. But the idea is simple. For any new pathogen, any new viral pathogen, or it could be essentially any pathogen, clearly you'll have some number of people who encounter that pathogen and survive and presumably develop a good antibody response to that pathogen. So simple idea is, what if you could take their blood and perhaps purify to some small extent the antibody fraction that provides, that allowed this person to recover from the disease, and could give that, in this case, convalescent plasma to a patient that's either is sick with the virus or with a new pathogen, or is at high probability for getting really quite ill. And so this idea, took hold pretty early in the pandemic. Really remarkably, a number of groups across the country were able to, do some limited numbers of trials. But very rapidly, this became a very politicized issue. So the FDA granted an early use authorization in August of last year for convalescent plasma, and subsequent data showed that really it's, at least in the trials that have been done, the efficacy was really quite limited. But it's still a great idea. You can imagine that, if there's great mechanisms to do this for the future, for new viral threats for which perhaps we might have vaccines that might take far longer to deploy, or other approaches that are far longer to deploy, having systems in place to deploy convalescent plasma is still probably quite worthwhile. Now, the other idea for providing passive immunity are monoclonal antibodies. And again, at the start of the pandemic, a number of groups beelined ahead to try to find individual antibodies And these could be, for example, recovered a single antibody that's totally an amazing antibody recovered from a patient that had overcome the virus, or found from some other mechanism. And if you could identify such a neutralizing antibody and manufacture it at scale, you could give it to patients that have the virus or at high risk from the virus. And again, this could potentially prevent infections. Now, there was a lot of work over the past year since the virus was identified to develop monoclonal antibodies. Many antibodies made it through early use authorization, but we anticipated several fundamental challenges with monoclonal antibodies. And the first of which is that because these monoclonal antibodies have to be given through the blood, previous work had estimated that only about 0.2 to 0.5% of the Cmax of the antibody that's in the blood would actually end up in the pulmonary spaces where the virus takes hold. So one could imagine that the amount of antibody that one would have to give to patients would be enormous. And that's what you really saw in these trials that read out last year, that that efficacy really came at really whopping doses, almost 8 grams of antibody per patient in order to see some sort of activity. And the other big constraint is that because these antibodies would have to be given through the blood, that would mean that patients that were either early in the course of infection would have to come into infusion centers or have to interact with healthcare personnel in some meaningful way. And that would just in general limit the broad deployment of this approach, because again, the capacity of doing that in the United States is really not that enormous, let alone elsewhere in the world. So the idea that we had was, what if we could come up with an alternative approach to passive immunity? And instead of delivering neutralizing antibodies through the blood, what if we could deliver them directly to the airways where the virus first takes hold, either from a little aerosolizer, a little device like this on the bottom, or perhaps in an intranasal spray, something like a Flonase, if any of you have ever used those. Now, the key advantage of an approach like this would be that because it could be self-administered, it would really dramatically open up the number of people that could be treated. So imagine if you've just learned that you're positive for the virus, you could immediately start taking an approach like this, and household contacts or other people that you've been in contact with could start giving themselves something like this without, again, having to go into, for example, an infusion center. The second thing is that because it's directly delivered to the site of infection, it could perhaps be more effective. And on top of that, the amounts of material required to get effective blockade would be perhaps far lower than the antibodies delivered through the blood. Now, while other companies had tried approaches like this, there's 2 key challenges. One is that one would really need an incredibly stable antibody-like molecule to do this, and that's because most proteins don't tolerate, going into an aerosol or being dried down in various ways. The other challenge is that because there's very few biologic drugs that are given through the airways, the pharmacokinetics and the other drug delivery approaches of this still remain a little bit unclear. So this is where my lab entered in. For many years, we've been using these really cool antibodies called nanobodies that normally exist in camelids like alpaca or llama. So before I go on, let me tell you a little bit about what was really neat about these antibodies. So my lab is also a structural biology lab. We deeply care about what the shape of the 3-dimensional shapes of proteins are. And on the left here is the 3-dimensional structure of an antibody that's flowing in your blood or my blood, and hopefully one of these you all have from the vaccine, a couple of these to target the SARS coronavirus. But antibodies are pretty complex molecules. They're made of 2 separate chains, and the business end of antibodies over here is what allows them to interact with basically an infinite range of antigens. Now, it turns out that camels and alpaca make these miniaturized antibodies called nanobodies, and those are derived from just this little domain of a conventional antibody. But they're remarkable because they can basically interact with, again, an infinite range of antigens. Now, on top of that, they're very small little proteins. And because they're so small, they're incredibly stable. So they're ultra stable. They can be heated up or dried down to a powder. And they're similar enough to human antibody heavy chains that they're not recognized as foreign by the human immune system. And finally, because they're such simple little proteins that are stable, they can be made in microorganisms like E. coli or yeast. And that means that they can be made at extremely low cost of production. Okay, so that's the general idea behind nanobodies. Our hope, again, was to find a nanobody that would act kind of like the normal antibodies that other groups were developing. But if we could develop a nanobody that targeted the coronavirus, perhaps you could deliver it via the airways. Now, the classic way of finding nanobodies is, you have your protein of interest, and then you go find a llama or an alpaca to immunize, and then that animal raises a response, an immune response for you, and then you go work up that individual antibody. that you can imagine is really quite slow and takes some time. So one of the technologies that my lab has been developing over, again, the past couple of years is a completely in vitro platform to rapidly discover nanobodies. So the idea here is that we take inspiration from a cool looking llama like this, for example, but instead of trying to go find a new nanobody from an animal, we essentially have created a very large library of about 2 billion unique nanobodies where these nanobodies are expressed on the surface of yeast. So the idea here is that we have a pool of yeast. In this case, it'd be 1 billion yeast. I've just shown about 10 or so here. And each one of these yeast has a unique nanobody on its surface. And then what we do with this very large library is go fishing for a yeast that has a nanobody on its surface that binds to our antigen. So in this case, we went into this library, the start of the pandemic, right about March when everything was shutting down for us. And we sorted these yeasts looking for ones that would bind to the spike protein of the coronavirus, discarding all the other yeast. And we iterate this process in about 2 weeks. In the following about 3 to 5 weeks, we ended up identifying about 21 nanobodies that had this property that they would bind to the coronavirus spike protein, but prevent its interaction with the ACE2 receptor. And then with a series of structure-based methods. So again, one of the really driving technologies for us has been the ability to see the proteins that mediate coronavirus biology directly using advances in cryo-electron microscopy. We could use these to very rapidly engineer our nanobodies to very, very potent molecules, to picomolar potency. And then we could link these nanobodies together to make them exquisitely potent, so potent that we actually can't measure effectively how tightly they bind. And that's really to driving towards femtomolar potency. And then finally, we could humanize our molecules to prevent the immune system from recognizing it as foreign. So let me just give you a quick highlights of what we ended up getting. This is, again, a 3-dimensional structure of the spike protein. As Barry indicated earlier, the spike protein has been characterized by people like Jason McClellan to be a very floppy protein. This little domain kind of moves up and down the spike protein. It's really important for how it functions because in this up conformation, it can bind this ACE2 receptor. Now, this nanobody that we found, which we've dubbed AeroNab for an aerosolized nanobody, binds, again, right on top of the spike protein. Again, this is an experimentally determined cryo-EM structure. So the nanobody sits right on top of the spike. And the way that it prevents ACE2 from binding, we know, is that it sits right on top, right where the ACE2 protein would bind. But what's really neat from a mechanistic perspective about this nanobody is that not only does it bind and prevent the ACE2 receptor from binding, but it reaches over to a neighboring subunit of the spike protein and basically locks it into this down state. So it has a 2-for-1 mechanism. It both prevents ACE2 from binding and also actually prevents the shape-shifting behavior of the spike protein. It's so crucial for its function. So with access to these structures, we could, again, do a lot of engineering that I won't bore you with. But I'll say one of the really cool things that we could do is link together these 3 little nanobodies that we could see in the structure as little beads on a string. So basically link up this guy to a neighboring one to a neighboring one to make 3 nanobodies on a string that would just sit down on this spike protein. And what we found is that we could create molecules that are exquisitely potent at neutralizing the virus. So this is a simple viral infection assay in a cell model. This is using Vero E6 cells. And what we could show is that this our final molecule, a molecule that we call AeroNab 6 × 3, inhibits viral entry with picomolar potency. And we think in this case, we're actually bottoming out the sensitivity of this assay because these molecules are truly femtomolar in potency. But on top of that, these molecules are incredibly stable. We know that they can resist heat denaturation all the way up to 60 degrees. And we can aerosolize them and they still remain completely stable. We can also completely dry them down to a powder and they remain completely functional. So the idea here is, again, we have an incredibly stable molecule that neutralizes the virus with exquisite potency. And what we've been working on for the past couple of months is finding a way to shepherd this idea, across the kind of classic academic translational valley of death. We found a great partner, a commercial partner who's helping us navigate some of this and is picking it up. And we're hoping now to do the kind of IND enabling studies to move it into clinical trials soon. But even if an approach like this isn't necessarily finds high utility for the current pandemic, we think certainly for future pandemics, having neutralizing agents that are just delivered directly via the airways could really have a big impact. So with that, I'll stop and really highlight this. There's an amazing team of trainees really who led this effort. Again, a big collaboration with Peter Walter's lab here at UCSF, led by Michael Schiff, Brian Faust, students Ruben Saunders, postdocs Smriti Sangwan, Veronica Rezelj, Nick Hoppy, Christian Billisbola, and Margan Boone. And again, all the structural biology work was done by this great consortium that we set up at UCSF to really rapidly get structures of many proteins for the coronavirus. So with that, I'll stop and take questions, and thanks again.
Thank you so much, Aashish, for a fantastic talk. First question from Nick, can you please speak in a bit more detail about what challenges you're facing in your translational value of death?
Yeah, great question. So I think the fundamental challenge is that the vaccines, it's not really a challenge, it's that the vaccines have done so well, right? So I think what we encountered, last fall after trying to find good partners for an approach like this was a concern that the vaccines would be relatively effective, and approaches like this would have relatively moderate utility compared to the investment required to get them, certainly into maybe not into clinical trials, but certainly past, for example, a large phase 3 to look for efficacy. So I think it's really one of finding the right partner who kind of understands the long-term vision of not necessarily being having utility for necessarily this strain for this pandemic, but perhaps future strains that are harder to tackle with vaccines, or perhaps for future pandemic preparedness. And that's, I think, what took us some time.
All right. And how often do you have to dose for this to be effective?
Yeah, here we're very enabled by another group, an industry group called Ablinks, that have been developing similar aerosolized antibodies for respiratory syncytial virus, which I think was earlier mentioned again by Barry. And what they've found is that a very similar molecule could be dosed once a day and have pretty good exposure in the lung. So we think an approach like this, again, would have to be taken once a day. But what we imagine, an approach like this would actually provide is kind of an adjunct approach, right? So perhaps an extra layer of protection for patients that either can't get the vaccine and have other comorbid conditions, or for whatever reason are not willing to take the vaccine.
Mm-hmm. And do you have any idea about like roughly the cost per dose that you would have with this, because it's an antibody?
Yeah, so there's what I'm slowly learning is there's a cost to produce something, and there's a cost of really doing it in a manner that that's safe to actually deploy to people, right? And I think the cost, actually, the dollar cost to actually make the raw goods is relatively quite low. The relative cost of doing the appropriate QC from batch to batch and knowing that it's safe is very different.
Mm-hmm. Right, because I'm just thinking along the commercialization paradigm, this preventatives have a hard time, right? Because you basically have to dose daily in the flu season or something. Although your approach could be done with a cocktail, right? With inhibiting several viruses probably simultaneously. So I'm just trying to wrap my head around that. How much of a difference it could make, how expensive it will be. For people to buy, or for insurers to reimburse, for somebody to take, over prolonged time potentially, right?
Yeah, and those are again all parts of the consideration, that we're slowly getting up to speed on. What I'll say just briefly is that another group previously has found single domain antibodies that are broadly neutralizing for basically almost all strains of influenza, and again, approaches like that could be great for again, pandemic preparedness, having those things, off the shelf, ready to go for whatever the next, influenza pandemic is going to be.
Exactly. And question from Anders, have you tried the different variants? Do you see any differences?
Great question. So we're intensely working on that right now. What I'll say is that we, in our preliminary work with the South Africa variant, we see decreased efficacy, and we're working on trying to basically create a version that's quite potent at the South Africa variant. We haven't tried a lot of others, mostly because we're particularly concerned about the E484K mutation that is again popping up in many variants of concern. So from a structural perspective, that's the one that we're most concerned about and trying to engineer around.
Mm-hmm. All right, and then another idea: could we use these aerosols to clean the air outside of the body?
We love this idea. I don't know how one shows efficacy for an approach like that, but we love it. Peter and I talked about coupling these with air purifiers or things like that to really decrease COVID burden in space, especially now that we know that, the primary mode of transmission really is aerosol mediated. But yeah, we love the idea. I just don't know, who the right person to talk to for that kind of thing is.
Right. There's been a bunch of companies also developing, UV and other technologies, right, to basically recirculate air. Even I saw a project with like direct solar tunnel, like that actually worked very well in low resource settings. Intense sunlight, they basically circulated the air through that. So it's a lot of innovation in that realm too, and it always gets me to think, why didn't we look at that earlier? Flu has a lot of morbidity and also mortality, and also do rhinoviruses. We could clean the air in like airplanes a lot better. But it took coronavirus to actually really push this innovation towards market. All right. Any other questions? I think that's it. Thank you so much, Aashish. It's been a pleasure having you and hearing about this interesting project. And I'm so glad to hear that you have found a path forward. Inhalation is also complex dosing form, right, for development. But there's a lot of advice out there. All right.
Great.
Thank you so
Thanks
much.
so much.
So with that, we'll go into a panel discussion with our speakers. So I just open up the mic for whoever wants to ask a question to the speakers who are still here. Please go ahead. All right. So, the questions we want to discuss mostly is what are the lessons learned and how do we implement change? I think there's a lot of elements that came out today that could be changed. I'm thinking the thought that forms most in my head is this idea of declaring a pandemic a true national security emergency, because that suddenly opens a lot of doors and cuts through a lot of red tape. Because to me, it is shocking to see how the research is still being treated like it's just somebody's hobby project and it's completely irrelevant for the wider population. Like in terms of the paperwork, in terms of the funding, in terms of the red tape. But really, we should remove all the unnecessary obstacles and make clear this path for innovation.
Yeah, I second that, of course. I also I feel like it's a lot of red tape in terms of pushing therapeutics specifically, as opposed to vaccines. It seems like an over-reliance on the vaccines as the cure-all, so to speak. I think there's a lot of value to the therapeutics, and that should be getting equal attention in my view, because I think it's a really parallel approach with the vaccines that will help end this pandemic for sure.
Right. Does anyone know though who actually sets the biosafety level for the new pathogen? Like I was trying to find that out. That wasn't immediately obvious. It's like a CDC guidance, but is it really a hard and fast rule, or is it optional that could you say, I'm doing BSL-2 with BSL-3 precaution? I guess we'll find that out in the workshop tomorrow, maybe.
So like for arboviruses, for instance, there's like a specific committee that decides on the biosafety level of like West Nile and yellow fever, and so that's like a whole, they're in the people who decide, but I assume there's also, Jana, do you know this at all?
No, we go by our permitting organizations and our BUAs, and in general, we just do everything at BL2 plus, or we don't do it. We do it through a collaborator. And most of the things that we need to do, we can do that. We can either deactivate or,
Yeah, I'm just wondering, because where is this committee? Is this at the CDC or is this locally at the schools? It's at the CDC, right?
We do both, yeah. We do CDC and USDA.
Ah, okay. Yeah. because I'm thinking, you know, for this, for example, if at least the private companies specializing in virology could have, you know, just started full blast looking for therapeutics really early, that would have accelerated things. But I mean, they first had to build the lab.
Yeah, but we have been able to get blanket authority, basically like 25 viral families. So it makes it, because we are looking for the unknown, it covers the knowns and the unknowns. Yeah. So you can get broad ones, but it's not easy. They come inspect every year and all that, but you can do it.
You can do it. All right. Yeah, I think part of this is also, my sense is, you know, you have to be a real specialist in dealing with this stuff. I mean, there's like ways around it and ways to deal with it that are efficient. But if you're just starting on a research project and you're coming from a technology angle, I mean, you're just clueless. I mean, you can't navigate, right?
Yeah, true. And I'm starting to, actually today is my first day of a new position on our campus at UC Davis, Vice Provost of Grand Challenges, in order to help the faculty basically to take on some of these new big things and have some support to say, like, how do we even approach it? Because I think for a lot of faculty, mostly junior, but I've heard from my colleagues that are really, you know, senior impressive faculty, it's just too much to take on. It's too much to deal with. I can't, you know, I'll just go in a different direction because of the paperwork, and it shouldn't be that way. We should be able to, you know, you had so many examples of that today. We should be able to figure out how to navigate this better and set up systems that support the teams doing things that help to save the world, right?
That is well said. It's well said. I think I fully agree. I mean, if I look at the scientific community on coronavirus, it's been so amazing what people have pulled off, you know, and how much innovation has happened and new technologies entering the virus field. I just feel, you know, from a national perspective, this is a resource, right? These people are a resource we should be using to the fullest. We shouldn't really bog people down completely with administrative overload just in case somebody ever sues 5 years now, or I sometimes can't even understand it, you know, why it's so complicated. And that's why I'm thinking we should, you know, tomorrow in the workshops, we also have some ideas to develop some, you know, particularly around regulatory, some crisis standards of care where you could, you know, focus on the important elements. I mean, we all want assays that work and that are precise and accurate. We don't want to tell people you have a disease that you don't, and vice versa. But there's elements in there that are, I mean, totally overkill and not needed. And those we could take out in a pandemic case, make things a lot easier, you know. And what you were just saying, I like this, that you now helping the faculty at UC Davis navigate, because in the diagnostic testing space, a part of the problem, if you really drill down to, is also the fact that they submit protocols, testing protocols to the FDA that are overly detailed about lots and manufacturers and things for every little thing. And FDA actually doesn't require that. So they like, it would be, so strategically, it would be much better if you submitted kind of a less detailed protocol, so you have more flexibility if you need it, for example, to take a different kind of swab. But once you submit, I'm using that manufacturer only, then you're locked in. So it's also a bit the navigation, you know, how do you navigate the system? Do you do it as detailed as possible, or do you think about, maybe I want to have more freedom and flexibility as much as is allowed, right?
Yeah, and fingers crossed that we can help, right? Because we always will be working within all regulatory compliance and all of that. But I agree with you. That's what we found with those more generic coverages, that you have to write it as vaguely as is acceptable and appropriate for the regulators, and they'll tell you if they want something more, and that way you have a lot more flexibility. Because yes, if you lose a vendor or something and you have to go back and do all of the paperwork over again, and you get a new group that's reviewing it, because those people turn over, in my experience, very frequently, and might have a whole different experience. So
Right. And maybe we need to think about, which elements are really critical and which ones aren't, because it's been such a, ultimately with executive power, the governor can do anything and kind of relax any rule, but it seems sometimes a bit random. We make it easier here, but then what really holds it back is over here, and that's not addressed, because it's the complexity of, for example, the regulation of diagnostic testing is just mind blowing. I mean, it's even, I poured over it for 2 weeks. At the end, I couldn't really tell you. I mean, I was able to find some things that should go, but I couldn't really tell you how to make it easier,
Yeah, that's why I think we need to rethink all of this and set it up in advance to be much more streamlined, and really, you can get pre-authorized and then go into emergency authorization. And I think we have a much larger voice than sometimes we think. So during the crisis, you have basically no voice. We know that. But ahead of time, academia is very well respected if we use it and bring it in a solution-oriented way. And we just often don't think about that and do it ahead of time, or we're not invited, so we don't know that we have that voice.
Right. And also the right people need to be part of the discussion. And I think it's sometimes I would say at a very high level also within science. And then where the rubber meets the road, it's like they have to talk to these clinical lab technicians, what they really face every day. And I'm sometimes afraid that's not happening enough, because this is really where the implementation is, right? It's such a public health idea, implementation science, but so important for this crisis and also for pandemic preparedness, because some of the stuff you just won't believe what the problem really is at the end of the day. And also, Eva, I mean, the surveillance studies is such an important thing. I mean, that should come in from the very beginning, right? Like a plan.
Yeah. Well, I had another thought actually. The surveillance study. I was just thinking, Jana, when you also think, but kind of from a larger perspective, and how to prepare and what kind of networks can be put in place in advance. So I noticed that there were some universities that were, really facilitating research, and that, where it was, everyone was functioning, and then there were others where we really weren't, and then there was kind of everything in between. And I understand that there was, like, at the local level, certainly, people wanted to do the regulations properly and have every BUA and everything taken care of. And then there was also, like in our case, it wasn't just UC Berkeley, but it was Berkeley, City of Berkeley Public Health, which was, kind of, I have to say, shout out, like they did an amazing job, right? We had like incredibly low numbers of cases and prevalence of antibodies, and now like giant numbers, percentages are vaccinated. So like, as like kind of handling public health, they did a really good job, but they were super restrictive with the campus also. And I think overly so. So I feel like there was kind of overly restrictions both at UC Berkeley and at Berkeley, but public health, but I also understand where that's coming from, right? It's coming from an unknown pathogen and wanting to, be, safe. Right. But there was like a huge heterogeneity across the country. And also ways that universities handled. So I was just thinking, I don't know, I won't be able to make tomorrow, Julia, but one of the brainstorming things could be potentially how to like create functional networks and some committees among universities in this country and potentially abroad about what, how to respond, like what's okay, what's not okay, what are reasonable levels of biosafety and what is kind of over the top, et cetera, so that there's a little bit less like dramatic heterogeneity in how, some universities and places are functioning, and then there's like completely different, and yet it's the same. I mean, just the fact that you're associated with a hospital, like everyone that was associated with the hospital, all universities were functioning at like 100%, but others, when you weren't, but that doesn't make sense because it's really the same labs and the same pathogen and the same research, so Jana, maybe you can take that on. But I mean, yeah, I just thinking, because there's got to be a way to make like the academic community be able to be more effective in the way we're all ready to do it, right? It's just a question of, having our universities being.
As you're saying this, I'm realizing though, I think it's also about the value of research, right? I think wasn't universally understood that research is part of the solution in this pandemic, right? It was like the value wasn't assigned. It's just like the same value like teaching a 200-person class and then you just shut it down. But the fact that, we're developing the cures and the diagnostics and we're learning about this new pathogen, I mean, we have to be allowed to do this work. And in fact, we should actually be supported to do this work.
Well, I think there was, I mean, like the whole IGI, like that pulling together of the campus was incredible. There was, I mean, Anders, you were part of, Dan, I mean, a lot of, that was amazing. And the scientists, the faculty rose to the occasion.
Right.
And it was incredibly, and everybody was like practically funded within 2 months, there's just a remarkable amount of work done, from IGI and from everybody. And then it kind of like ground like to the screeching halt when it moved kind of out of that kind of incredible phase into the next phase, if we should, but I know that there are, like I said, certain universities, and I've been in contact with them who were very like prescient and looking forward and had already kind of ways to facilitate it. And I think that would be, great if that was more like the norm. And we can, look to those for guidance, or to come up with a way that is just more, so, I mean, because you're talking about that diagnostics and like public health laboratories, but I also think we could apply the same, kind of what for the next time to research at academic institutions as well.
And for this IRB thing, I'm wondering, so who regulates this? Is there any central unit that just says, certain type of study is now approved blankets, or is it truly at every university and they just totally independent?
It's pretty, I mean, there's certain I wish Lisa had to run to another thing because it was running, but I mean, honestly, there were certain studies that were identical to ours that at other institutions were considered public health and didn't even get reviewed. Like they were just considered, if something gets labeled public health, and if it's therefore not research, then like apparently, and it was like the same thing, and they didn't even go through IRB. I mean, the IRB decided that they didn't need to review it because it was under the dominion of like public health, so that was really interesting. But I mean, there are certainly, NIH has the Office of Protection of Human Subjects, which is kind of an overarching, but then every institution, follows those guidelines, but they, I think, interpret them, differently.
Right. Yeah, and I think one thing we've also seen on a psychological level is just the isolation, the anxiety, and people getting very risk averse also in all their decisions, right? Somehow, particularly if they have authority and responsibility for safety, suddenly it's like, it's kind of, I don't know, it's a psychological thing also.
But that's why I think having, and people did reach out to each other, but it was much more informal. People were like reaching out to all of their contacts, to gather information and et cetera, as opposed to having like a ready-made network that was able to respond in a more kind of coherent fashion.
And why was your study not considered public health?
Well, because I can't answer that. Why the I mean, like I said, it would be great because actually Lisa actually sits on the IRB also. I mean, that's actually one of her university extra functions, and so she was in this really interesting position.
Yeah, navigating.
Between it, but yeah, I don't know. I just heard that others decided.
Usually, if you say in advance you're planning a study for publication, then it's not exempt in the same way as if you say I'm in the middle of an outbreak or public health response and doing diagnostics and others. It doesn't always hold, but that you doesn't mean you can't publish. But if your goal is putting together manuscripts for publication versus assisting the response, I think they use that filter.
They could, but in this case, that wasn't
right.
Doing this.
Right.
It strikes me that the protection of patients is a high good, right? You want to make sure, but at the same time, if the benefit from the study could protect them from the pathogen or from the outbreak, right? The whole balance is shifted a bit in terms of what you should approve, right? Because it's not just a risk to them with a finger prick or whatever you're doing, but it's also a benefit. So it's a different calculation. I'm just wondering, you know, it's surprising to me that they look at it that carefully for these non-invasive studies, really, and it's so difficult, right?
Yeah, even some of the work that we had through Predict that was IRB approved, and we had a blanket IRB for all countries, and then we did an IRB internally in each country. Then later when we went to publish the data to make it public and de-identified everything, we still had the entities publishing and the funding agencies come back and ask us to limit the data even more for HIPAA changes that had happened since we got IRBs, and really make it almost unuseful, the data, honestly. And so we, you know, you have to fight back and blah, blah, blah. You know, it's like, do we want the data to be useful or not useful? There's no way this could hurt someone. You know, we've already been through IRB for that. But yeah, it's tough.
It's tough, yeah. I mean, it's just, I don't know, being in this, witnessing the situation all around me, I think we don't talk enough about the toll it takes on investigators, you know, because on its own, you think this is okay, another administrative process, you know, we can do it. But ultimately, if you're in the daily grind in the middle of the pandemic, you know, chaos, and you know the news is calling all the time for you. You have to do your work and research. You're one of the leaders in the field. You know, I mean, how much can a human being really do? You know, at some point, it really takes a toll on people. The sometimes insanity of this administrative system, and it kind of burns people out. I think it's you know, we have to be careful with that as a society, because you know, I mean, it's counterproductive if we want to get over the pandemic, right? That was just my personal sense of it, you know. But for another question I had for Eva is, with your blood spot method, you know, with all this stuff you worked out, I mean, the barcoding, the sample processing, is this something you're considering maybe putting in a package, like for the strategic stockpile or something? You know, because you
actually, this one thing is hilarious. This is so funny. The Smithsonian is putting together like a little something about the next pandemic. I don't know. They asked for one of our kits to put into the museum, which I think is hilarious. But anyways, in reality, what we've done is we were like, people were calling us, like, I mean, we were trying to actually just get the IRB approved or get the thing off the ground. And there was like 20 people a day practically like contacting us saying, hey, we want to do this. How are you doing it? And so we shared very broadly, you know, as soon as we had something that was like, okay, this is viable and we can get this approved, you know, there was you know, with UCSF and Davis and I mean, a lot of different places, including a lot, you know, Lisa in particular, she's trying to get back on, but she's in another meeting, but she, you know, with the California Department of Public Health, there was a whole series of you know, of discussions, and we were kind of keeping each other abreast of the surveillance that was going on. And like I said, now they're taking off and doing, you know, a whole statewide surveillance. And so we just tried to make anything that oh, there you are, Lisa. I was just talking about like how can we have others benefit from everything that we've learned from the sero surveillance, et cetera. And I was saying that, you know, our work with the CDPH and with many other campuses and other groups, just kind of sharing protocols, sharing how to do it. And now, like, you know, there's all these silly details that absurdly took all this time, but like the little business card with like how you drip your blood on it, I mean, that was really useful, you know, and so that's good. Like, I think there's right now, I really feel like we've, you know, come to a place where there's like a feasible, viable way to kind of do this.
Yeah,
and you know, have interest companies in doing it. They're absolutely want to do this for like the next time around, you know, with the use case and understanding now with Mike Bush how we do, like, there's different kinds of antibodies given the different formats, which was not appreciated. They're going to submit a paper soon with 21 assays compared in longitudinal sample sets that shows very clearly, you know, some of that aspect. So I think that we'll be way better off in terms of understanding what kinds of serological assays, you know, work for what kinds of questions and what kinds of samples. And I think that is going to be way further along for the next time around.
Because I think if the work you've already done, also with the communication and the videos and, you know, what works with teaching people how to take the blood, I mean, if you can put that in a kit or something, and we could have that ready for next time, or publish, you know, as a method or something, that would be great to have. Right.
Lisa, anything from you?
It sounds like you covered most things.
Yeah.
Well, no, but we are. I mean, like that's.
Yeah.
I mean, we have a whole, if it ever materializes, but there's a paper just on this, the whole kind of validation and all of the things for the silly DBS, you know, et cetera, et cetera. And I think the kind of bringing to scale was another aspect of it. Not like, I mean, 18,000 is not scale in relation to the whole country, but it's more than like, you know, a pilot that shows that, you know, you can do it in 50 people or something, you know, so, but yeah, of course, anything that's useful is good. And kind of getting it out there. Lisa, there was a question about who determines like what level of IRB is needed for a review. I mean, because remember how there were some places that didn't even consider the studies like needing IRB review, and it was just public health, whereas that was a very different perception here. And you're on the IRB, so who decides this?
Well, we have other people here too that are on the IRB, I think, maybe. Well, I think it gets decided when it's submitted when the protocol is submitted, and there are different categories that are used to determine what kind of review it gets. And ours had all of the, somebody mentioned earlier, you know, it wasn't surgery, but it also had there were a lot of issues. One of them was the big unknown around the in-home or the at-home testing. So the IRB, you know, whether or not you have an investigational device, is it FDA approved? I mean, it turns out that everything that was in our kit had to be approved by the IRB because it's either something that's been FDA approved or it's not. It's considered an investigational device. So even a swab or something like that, because we were not using approved reagents, approved take off the shelf type kits, and we weren't doing things in a CLIA way. All of our work was done in CLIA labs, but it was not according to CLIA protocol.
Right.
So yeah, there were a lot of complications. The IRB was great. I mean, they were so supportive and they worked as fast as they could to push things through. There was just, it was complicated, and you know, the expedited review for such a complicated protocol needed, you know, more hands, probably, you know, immediate review by an MD might have helped. I've talked to them a lot about it, and yeah, I'm on the IRB, and I have been part of reviewing a lot of other COVID study protocols that also required, you know, many back and forth. So I think that the tough thing with the IRB too is that in some ways, if they had enough staff that they could have conversations with you real time, you might be able to work out some of these things before you get into the system. And then there's a lot of back and forth administratively. So that really does take time.
All right. And who gives the guidance for the university IRBs? I mean, if somebody would say we have this global pandemic, let's make, you know, at least the lowest risk cases, we'll just give blanket authority. I mean, the head of the IRB is Rebecca Armstrong. And then the head of the study section, sorry, not the study section, but the review committee. The IRB has 2 review committees, and it depends on the type of research that it is. But the head of the committee that would approve the kind of protocol that we submitted is Bill Jagist. So those are I've talked to Becky about this at length, you know, what could be done in the future. I mean, for them, like anyone, it was all just, you know, overnight urgency. Okay. But these are all in Berkeley, right? These are not national institutions at NIH or something.
No, I mean, this is local. Berkeley has their own IRB.
And they just operate completely independendly.
No, they have to answer to, you know, the federal government, FDA, they answer to federal governing bodies. And
probably the most efficient thing will be for the federal government body to say, you know, we have a global pandemic, we're changing the rules a bit, right, on the IRB approvals. For example, you know, it's okay to query de-identified patient data without, or do a survey. I mean, they even do things like you can't do a survey, IRB approval, things without patient contact, physical contact, you know, you could.
Yeah. I mean, ours involved so much contact and in-home things and all that, but the FDA regulations played a big role too, because the FDA probably a bigger role in many ways, because the IRB was taking into account, you know, what the FDA was saying at the time. And there was so much back and forth about what can actually be done at home. And the FDA didn't want anything done at home. No at-home virus testing.
Yes.
And that was huge.
I mean, they changed their mind on that, but it took a long time and a really bitter fight. Yeah.
Well, so Seattle Flu, which was a big project, which became Seattle Scan at the time of COVID, and they were doing a ton of surveillance. I believe they were responsible for figuring out the residential, you know, senior care home outbreaks in Seattle. I think a lot of that came from the work that they were doing. And then I think they had done something like 17,000 self-administered tests, and there's somebody injured themselves. They injured their nose. They were doing the nasal swab, but they went too far, and they got shut down. So they were shut down for that, and that was impacting a lot of other people trying to do these surveillance studies with virus testing. So, you know, but I guess the FDA was also trying to figure out what was okay and what was not, and preservative for the swab. I mean, it had already been shown that the swab test was way more sensitive if you preserved it in RNA DNA shield. And then there was concern about RNA DNA shield being in the home. And even though it's a really small volume, I mean, at the end of the day, that's basically the test that's done everywhere now is, you know, the swab and the preservative. But we had over 16,000 kits with RNA DNA shield in the home, and we had, knock on wood, no issues, because we gave, you know, it's a tiny, tiny volume, and we gave really strict instructions. But it was things like that. And the FDA was reluctant to say that was okay. They did not want that. They wanted people, if they were going to do anything, use a dry swab. And then the dry swab had all kinds of other issues because Berkeley's EHS didn't want dry swabs to be received. Anyway.
And it was so good with the FDA, and I'm like, you have like Drano, you have like gallons of Drano in like under your sink, but like 0.2 mils of this stuff is, you know, with like all this, it's just so ridiculous.
It eventually all got worked out. But the problem is that the timing of all of it, it was a huge, huge time sink. It's all worked out now. And, you know, there are FDA approved at-home tests for
I just talked to a guy who was involved in developing some of those, and it was so interesting because he knew all these players in this game, and he said most of them got shut down, and the one who succeeded was one who strategically used an FDA-approved saliva collection device. So just because you spit in that tube and not in the other tube, because then the FDA couldn't say no because they already before.
But they were expensive.
That spit collector.
Well, there were problems too with the saliva, depending on which lab you worked with. And we tried to partner with labs to do the saliva testing. And it was difficult because different labs were having different problems, and robotics were having different problems with using saliva. We really wanted to use saliva for virus testing, but it turned out that we couldn't. And the swab, I mean, the swab is pretty foolproof and has been used now. I mean, it's used everywhere. The thing is, if we get into a position where we need to do testing, like we had to do and are still doing, if we need to do that in the future, I think at least we're completely set up now where big sites like that are in existence now could just be set up and it could be done. But it's just, it was a very painful process.
John, I have a question for you. You know, with the PREDICT study, I mean, you've kind of done what we should do, I guess, on a national level, work with all these different countries, with stakeholders, you know, get everybody on board. So how do you do it? Like, what are your best recommendations for that, you know? Because the level of effective collaboration is pretty strong there, right? So
I mean, the way that we approached it was, you know, without expectation, and to just bring, here's what we think might be useful and how we can contribute, and who on, you know, are you interested in who in your ministries and countries do you want us to work with? So we were just very open-minded about that to the extent we could with funding and compliance restrictions. But we almost always could go back and make the argument that we needed to add or remove or replace partners. And when it was actually the first internationally based project I've ever done that that everyone said yes and they wanted to participate, and yes, please, please come, and when can we start, and let's do it quickly, which makes sense because obviously they wanted to get ahead of things, and that was nice. It's been much different. There have been a couple different efforts to try and start it here, and immediately gets mired down in, wait, whose jurisdiction is that? Wait, yeah. That needs to go there. No, this needs to go there. And that's what I was sort of alluding to in my talk, is that I'm already seeing those kinds of obstacles going back up, that people are, yeah, we should all work together, but this is the structure I'm in, and that seems like too much to take on. It's kind of like the climate issues and people saying, well, that's too much in the future, too far, that's too hard. So I think to make progress, we have to listen to that and say, okay, what's a small piece we can bite off and move forward with? And, I've been working with FDA organizations that never used to want to hear from us are now coming to us and saying, let's do this differently. So I think there's a moment in time, but I feel the window is really closing. And it's just a human nature thing. So, but being open and collaborative and trying not to take anybody's credit away or all of those things is just playing to human nature is useful. Again, that's why we need lots of disciplines. We need our colleagues who study that, right? What's going to be palatable? What's, how can we get started to do it even better?
Right. All right. Any other questions from the audience? Yeah, so with that, maybe I'll take the last 2 minutes and quickly introduce our workshops tomorrow. If anyone is available to participate or to jump in from time to time, we start staggered from 9 a.m. on, so 9, 9:30, and 10 are the starting times. And then we do this like a hackathon workshop we did a year ago, which is basically self-organized. We do an introduction in the morning, and then little working groups form. There's a Slack channel, and people do some research and work together, and in the evening come back with a small outline of what they found or the most interesting things they found, and then we present it together, and that can depending on how it goes, could evolve into a white paper or some publication of some sort. So we have 3 workshops set up. There's also possibility to do something new. If anyone has a burning idea that isn't listed, we could also add a 4th one if there's enough participation. The first one is about regulatory science and pandemic preparedness towards a crisis standards of care. This idea comes from emergency medicine. All emergency rooms have a certain setup here and have thought through what would happen if they had, a big disaster close to the hospital, like in our case, maybe a big earthquake happen and emergency rooms are overrun with people who need help. How do you triage? Who gets what resource? So they think through this when times are calm, and at least build somewhat of a framework of how to react, so that when patients pour in, they can focus on the patients and on the care. They don't have to make all these highly difficult decisions right in the moment. So what I'm thinking is we could envision something similar for regulatory science, because this clearly hasn't happened at all. We have like one system and we apply it no matter what. And the FDA is the only unit that has something like, has thought about it a little bit through emergency use authorization pathways, but CMS, to my knowledge, hasn't really done much. And as we have seen with this testing capacity debacle, we kind of held up by things sometimes that shouldn't be issues, or we could solve in a better way while maintaining principal tenets of assay accuracy, precision, and good interactions with, and ethical concerns around patient data. So same is true around HIPAA as well. So trying to develop some thoughts here, what could be proposed. So the idea is to think through this while you have bandwidth to do so, and carefully can study all this if there, and then once crisis hits, you don't have to do all this while you're trying to struggle with the outcomes of the crisis at the same time, which is what happened this time. So that's one idea. The second workshop is to, how to refocus research to a new threat rapidly, effective funding mechanisms and science administration to harness reserve capacity. This is about methods of funding. It's also about lab reopenings. How can we make sure that the researchers who have the expertise, which are limited in number, can actually put their best foot forward and do the work they need in order to get that threat under control. Also understanding that, the balance has to shift a bit. I think the safety versus benefit balance, if you have a pandemic, raging that kills people and we have no therapy, we have no vaccine, and in the beginning, barely any testing, the whole balance of evaluation of risk to a patient, if I do a finger prick versus just not being able to do anything against the pathogen, it's a different balance from just testing people for things where they can change their exposure or something like that. So that's about research capacity, the academic reserve capacity that's there and could be used better, I think. And the third one is a workshop to accelerate therapeutics development and develop sufficient high containment drug discovery lab capacity around BSL-3 and 4. I mean, this time we had BSL-3. In theory, one could get a BSL-4 pandemic. I mean, we hope that won't happen. But if that happens, we kind of totally host, because we have like 3 labs in the US who can even do that work. And for screening, there's only one at Southern Research, and I don't know if that's even still active. So there needs to be some thinking about infrastructure. I think politically the decisions were mostly made with envelopes of anthrax in mind. I mean, that's one lab per state and so on. But if there's something that is very transmissible, like this pandemic, or even viruses that could be way worse, we need to really think about how we develop countermeasures and how we can do this in a reasonably safe way in laboratories, what kind of infrastructure is needed. And that pertains both to personnel, people who are trained to work with pathogens like that, and also the lab infrastructure. All right. Well, with that, I want to thank all our speakers. Excellent presentations. It's been a pleasure talking to all of you and discussing this. And we hope we can gather elements from this from the symposium and put together at least a report, a conference report or a white paper with what has to change. I feel also like John, I think the window is closing a bit. I think after this pandemic is under control, which is not yet, but there will be a small window of opportunity to make our voices heard. I think before we go back to, just highlighting the positive and moving on, but I think some changes could be made, and I'm hoping we can use that opportunity. All right. Thank you so much for your time today and for your contribution. All right.